So, in this lecture I shall go through that how microfluidic system used for cell culture and how it differs from that traditional cell culture system used for 2-dimensional and 3-dimensional cell culture system. Furthermore, we also go through that what are the main challenges to face that microfluidic system to understand the fundamentals of biology as well as application aspects of the biological system, particularly healthcare related systems. So, why we need cell culture? Basically, the goal of the cell culture is to repeat or recapitulate the closely as possible as your microenvironment present in vivo system that will be replicated in vitro and with adequate some replica by which we can get statistically significant results in a reasonable amount of time. But balancing between these two aspects is not achievable in macro scale culture. Either you have to sacrifice the model accuracy or sacrifice the high throughput or vice versa. So, in that situation that microfluidics is likely to have a largest impact because it has the potential to improve that both model accuracy as well as high throughput simultaneously. At the same time, it will provide the freedom to tailor the microenvironment while also reducing that scale of that experimental platform. So, what do you understand by the microenvironment? So, cellular microenvironment is a dynamic in nature and it encompasses that biochemical cues means soluble factors, physical means cell interaction, cell ECM interaction and external mechanical and electrical fields, and physiochemical or that means pH, oxygen tension, temperature and osmolarity factors. That gradients of all these type of things also play a crucial role and these gradients act as signal themselves to regulate the cell function. What are the gradients? Say chemotaxis means chemical gradients, basically it is growth factors, soluble factors and chemokines and etc. Then haptotaxis means is a gradient of ligands like say receptors, addition molecules, collagens and their receptors. Mechanotaxis, it is a gradient of mechanical forces means shear force, extrinsic forces, ECM resistance. Then durataxis, that is rigidity of that extracellular matrix, the topography of the matrix. Then polarity means that is apical sites of the cell and basal side of the cells, they have a different types of biological functions. All these things will be affecting that your cellular signaling by which cell can grow, manifest their biological activity as well as specialized function for the cells. So, microfluidics is expected to play a significant role in the design and implementation of such stable gradients of various forms in the cell culture. What are the cell culture prerequisites? For the prerequisites, we need cells that may be primary cells or cell lines or immortalized cells. Then we need media for culturing the cells which will give the means nutrients and culture wares means flasks, that is plates 96 well plate, 12 well plate or T25 flask. Then carbon dioxide incubator which will provide that lot of functions, maintaining the temperature, maintaining the 5% CO2 level for the buffering activity and maintaining the humidity as well as. Then laminar hood or is a laminar flow cabinet, it is the essential part to maintain the sterility during the operations by which any contaminants cannot go inside that your culture system. Then we are coming centrifuge, it is a low spin centrifuge to pellet down the cells or suspended the cells to washing several times during cell culture experiments. And microscope, it is optical fluorescence mode, because we have to see the cells after culturing or after incubation, and most of the optical readouts in fluidic platform are the optical readouts. So, microscope is very important. Then that liquid nitrogen canister, it is used for storage of the cells, because we have to store the cells after culturing, then again revive for the next set of experiments. And water bath, it is just to thaw the cells after taking from liquid nitrogen canister, and at the same time if we want to make the culture media come to room temperature, we have to put in a water bath. So, what are the types of cells used in cell culture in vitro? In our system, most of the cells are adherent type, means they need some substrata to adhere, grow, then multiply, then manifest their biological readouts, then it will die. So, substrata is very much essential for most of the cells, these are called adherent cells. For the circulatory cells, which are non-adherent type or non-anchorage dependent independent cells, these are the say which cells are flowing in the bloodstream, RBC, WBC, etc. So, what are the primary cells? Primary cells are cells taken directly from the living tissue or a biopsy material and established for growth in vitro, and they have a few population doubling and passage numbers. So, like for example, this is a mouse, we are taking that skull tissue for isolating that osteoblast cells, but it is keep in mind if you want to do any animal experiment or human tissue samples, you have to take the ethical clearance from the institute or organization. So, after the ethical clearance, we can use this mouse, then anesthetized, and using that sharp scissor, we can take the little bit tissue of the skull, then it is making a piece chopped to pieces, very small pieces, then we can incubate with the trypsin solution for maybe 5 to 10 minutes to remove that ECM matrix, then pipetting several times to make it is a fine suspension, then you can pass it through a fine mesh to get that cell suspension, then you can centrifuge little bit to remove that excess trypsin, again wash it to remove that excess trypsin, or you can neutralize the trypsin using little bit BSA, then again suspended it. After suspension, you can check the viability of the cells by Typhon blue assay and count the cells by using hemocytometer. Then you can seed the cells in your petri dish by which cell will grow, and this is the primary cell lines, and this will grow, this has a limited means generation time. And what are the cell lines? A cell line is a permanently established cell culture that will proliferate indefinitely given appropriate fresh media and space. And immortalized cell line, they are only tumorous cell lines, they do not stop dividing, or cells that have been artificially manipulated to proliferate indefinitely and thus be cultured for several generations, means infinite lifetime until unless you are short of that nutrient media. So, this is your human cervical cancer cell HeLa, most usually in the almost all the labs. This cellular morphology is like this is very much immortalized cell line. So, primary cell lines, they have a limited lifetime, and this was discovered by Hayflick. So, this is called Hayflick limit, means Hayflick and Morehead started the potential human fetal lung fibroblast, this is a primary cell growth in vitro, and observed that after 50 generation the growth of the cell begin to slow down and ultimately leads to complete death of the culture. The number of population doubling level to human cells generally between 20 to 80, but it can be much shorter depending upon that cell type. Like say if you culture that HUVEC cell lines from that umbilical cord, it may go only 4 to 5 generations. So, Hayflick effect, there is 3 distinct phases. This is phase 1, phase 2 and phase 3. Phase 1 is the primary culture, phase 2 is the subcultivated cell during the period of exponential replication. This is the most important phase where you can culture the cells, and phase 3 is the senescence phase, means in that period cell replication ceases, but it is a metabolically active. Cells may remain in that state at least 1 year before senescence, means after that cell will die. This is the nature of primary cells, and primary cells are very good for experimenting because it has all the characteristics which present in the in vivo situation. So, if you want to grow that cells, we have to subculture it. What is subculture? Primary culture are generally subcultured at 1:2 ratio. They are split in half with each passage. Most continuous cell lines, means cell lines or immortal cells, they replicate at a higher rates and subculture at a much higher split ratio, means 1:4 or 1:6. What is passage number? It is generally the number of times cells have been subcultured into a new vessel and is used to know the age of the cell culture, because age is a very important factor to know that cellular behavior. So, what is population doubling time? PDL, it refers to the total number of times the cells in the population have doubled since their primary isolation in vitro, and it is more accurate indicator of the age of the culture than a passage number. Why? Because the number of cell doubling in a given growth area depends upon that cell seeding density, means in a given area if you use say cell density 1 into 10 to the power 5 per ml, say 500 microliter, then you can use 3 into 10 to the power 5 in the same situation, then you will be seeing that confluency very quickly for higher cell density and confluency is less for less cell density. So, passage number will not be the proper means way to judge the age of the culture. So, how will you determine the PDL, population doubling level? This is a very simple formula, 3.32 into log xc minus log xb plus s, where xb is the cell number the beginning of that incubation time, xc is the cell number that is end of the incubation time, s is the starting PDL. Means if your s is 1, then you can calculate PDL, then s is 2, then also you can PDL. So, in that PDL is the most appropriate to know that cell culture age, particularly it is used for stem cell culture, it is very much essential. So, if you culture any cell, this is a growth phase, basically is a lag phase, log phase, followed by stationary phase and death phase, and this is the pattern in a any stationary phase culture due to their nutrition limitations, that ultimately stationary phase they will enter, then death followed by death. What are the cell culture media? Generally we are using DMEM, means Dalbecke's modified Eagles minimum essential media. It contains that both micro and macronutrients. That media serves as a biochemical microenvironment of the culture and consists of essential amino acids, vitamins, salts, carbohydrates and other components in the aqueous solutions. The culture media should be supplemented with your calf serum or bovine serum to provide that growth factors, because mammalian cells they need not only that macro and micronutrients, they need the growth factors in the form of cytokines, chemokines or insulin like growth factors. So, natural source is that fetal calf serum, but the composition serum is not always constant, added in natural source, this is batch to batch variation will be there. So, you may not get consistent result with this method. So, nowadays people are thinking to use artificial media or serum free media, where that all the ingredients growth factors are supplemented artificially, because due to the advent of recombinant technology growth factors are available in large quantity. So, synthetic media are available for particular development of particular cell lines or maintenance of the particular cell lines, and this media is supplemented with antibiotics as a penicillin and streptomycin at the 0.5 milligram per ml to reduce the microbial population, by which external your contamination will not be there. So, you can get that liquid media from commercial source, or you can get that sachet that is in powder form. You can dissolve the powder form in 1 liter media or according to the specification. Then you have to sterilize the media. How will you sterilize? Generally we cannot use autoclave, because due to the presence of growth factor which are the heat labile. So, you have to pass through that 0.2 micron filter, by which filter will be 99.9% bacteria free. To check that sterility of the media, so you have to put that whole media in 37 degree centigrade incubator for at least 24 hours. Then after that, if you check that media color is red, means phenol red, it is okay, and there is no turbidity, means it is fine. If turbidity is there, then you have to find that there is a microbial contamination is there. So, you have to discard it, means everything you have to do so much meticulous that no contamination should be there. What are the physico-chemical properties of cell culture media? That is pH. pH generally in between 7 to 7.4. Phenol red is commonly used for indicator. It is red color in 7.4 and yellowish color in 6.5. After the when that culture becomes acidic, it will be yellowish, it is pH will be 6.5. And that pH is maintained by carbon dioxide bicarbonate buffer, that is why using CO2 incubator and maintaining that 5% carbon dioxide level, and you can use HEPAS buffer also. Osmolarity you are maintaining generally in between 280 to 380 milliosmol per liter, and oxygen level is very important, because oxygen consumption rate depends upon cell to cell. It varies between 0.05 to 0.47 millimole per liter per hour for 10 to the power 6 per ml. But for stem cell culture, oxygen requirement is little bit less. So, you have to maybe maintain that oxygen concentration is less. Temperature you have to maintain 37 degree centigrade. All these things means at least pH, osmolarity, oxygen levels, temperature and humidity, everything is maintained using carbon dioxide incubator. Culture ware is a very essential part where to culture the cells, and in traditional system we are using that polystyrene based petri dish or flasks oils, 96 oil or 24 oil plates or 12 oil plates, and these polystyrenes are hydrophobic surface and typically plasma treated to make it hydrophilic to facilitate the cell adhesions. For certain cell types you need to treat the surface with extracellular matrix protein like say collagen or fibronectin, or you can treat with polylysine, coating the polylysine, by which cell can adhere nicely, and then they do their all the biological activity starting from growth to manifestation of biological readouts. So, the key factor of cell culture is that the viability of the cell during regular maintenance of the culture, including appropriate seeding density, regular change of culture media, monitoring the cell growth and timely subculture. Take for example, this is a 6 well plate and its diameter and heights are respectively 35 millimeter and 17.65 millimeter. Growth area is around 9.6 centimeter squares. We can seed the cell 0.3 to 10 to the power 6 cells, and we can give the media 1.25 ml, by which you can get that media height 133 millimeter. Why I am telling this? Because if you give this media, we will be getting enough head space by which that oxygen can diffuse to the media, and there will be no oxygen limitation, because polystyrene is not permeable to oxygens. Cell number will confluence to 1.2, means 4 times you can get cell numbers within a say 70 to 90 hours culture, but you have to change the media after 48 hours. Means if you look at the 48 hours or 24 hours solution, that cells means color becomes red, but after 48 hours that media color becomes yellowish, means it is acidic. So, it is the proper time you have to change the media, because of due to metabolism lactic acid or some acidic products are accumulated. So, is the proper time to change the media before the confluencing of that cell culture flask or plates. So, basic steps, how will you culture the animal cell culture in a normal laboratory situation. So, normally what happens that cells are stored in a liquid nitrogen container, that storage media is 5% fetal bovine serum, 40% incomplete media containing 10% DMS. And we have to take all the precautions. So, before doing the cell culture, so you have to wear the surgical masks, surgeon's caps, nitrile gloves and washed aprons. Then wiping hands with cotton swabs soaked with 70% isopropanol ethanol. Then you are entering the cell culture room and sterilizing the lamellar air by flow cabinet with 70% alcohol and UV radiation for maybe half an hour or 1 hour according to the requirement. Now, you take out that cell storage from the liquid nitrogen can and immediate thawing using that water bath at 37 degrees centigrade. Then dispense the contents in a 10 ml complete media and centrifuge at 1500 rpm, maybe 2 minutes. Now, you discard the media, resuspending the cell pellets and fresh media, dispensing cell suspension in a culture flask or microfluidic culture furnace anyway. Then culture the cells in a humidified cell culture cabinet as I mentioned earlier at 37 degrees centigrade, 5% CO2. Then you have to check the cells every 12 hour or 24 hour how it grows or how it sits, then look for it. Then how to maintain the cell culture. So, after growing the cell in a culture flask maybe after 72 hours, you have to, for adherent cells, you have to take out the cells from the cells, because as it is adhered to the your surface of the polystyrene disc or plates. So, how do you remove? So, for that you have to use trypsin EDTA solution, is commercially available. So, after that dispensing the spent media from the culture flask, rinse the culture vessel with trypsin EDTA solution for adherent cells, and you may be incubate for 5 minutes or maybe less than that according to the cell line to cell line, and neutralize the excess trypsin suspension with twice or thrice by some volume complete the media, then collecting in a complete centrifuge tube and spinning it. Right now your cell will be pelleted down and discard the media again you give 1 ml media, counting the cell in a hemocytometer. Then you can do the experiment passaging, then you can storage the cell for further use. So, now we are coming that microfluidic cell culture, because up to this is same for normal cell culture system and microfluidic cell culture system. So, in microfluidic cell culture, if you want to do, means we shall be doing it, some requirements are there, means choice of material for device fabrication, and the geometry of the dimension of the culture region, and the method of pumping and controlling fluid flow. So, what material we are choosing for fabrication purpose? Generally, polydimethyl siloxane or PDMS is a silicon based elastomeric material is mostly used. PDMS is fairly cheap, easy to mold, ideal for rapid prototyping of microfluidic designs, transferring micro patterns with high fidelity, non-toxic to cells, and it is a gas permeable. Excellent optical properties is very much essential for microfluidic cell culture system, and including low autofluorescence and optical transparency for imaging applications. Generally, we are using that PDMS, that Celgard, it is supplied by the Dawex company, 10:1 ratio base is to curing agent for that purpose. It may be vary, but generally we are using in that ratio. To make enclosed microchannel, PDMS can be bonded to different materials, maybe PDMS itself, glass, polystyrene, and it is easily done by various methods such as oxygen plasma treatment and additional curing method. But PDMS has some drawbacks also. It sequesters a small hydrophobic molecule like say estrogen or hydrophobic molecules of molecular weight less than 500 Dalton, and leaching out of the uncross-linked oligomers from the PDMS bulk during culture, and that may affect the cell culture system. But if you culture the long time in cell culture, this leaching out PDMS which are uncross-linked they can affect the cellular growth, biological readouts and biological functions. So, we have to take precaution for that. So, what are the alternative for this? Alternative is maybe polystyrene or silicon, but polystyrene is very good material. It is traditionally used all the cell culture system, but molding to the desirable means shape or geometry, it is not user friendly like PDMS. Glass is versatile material. So, we can use, if PDMS is making problem for your cell culture things in microfluidic system, we can use polystyrene or glass, and many companies are coming with this concept. So, what are the geometry of microchannels? Microchannel dimension for cell culture is typically in the range of 100 to 1000 microns for the study related to population based analysis. Means in that channel you can use 100 cells, 200 cells, but it is much less than that of traditional cell culture. And important geometric consideration in microfluidic channels is the height to width aspect ratio, especially in PDMS based device, because that PDMS device, if you use that integrated valve, as it is a flexible, that may be buckled basically. So, that is one of the important aspects you have to take care of. And you can make that tailor made various types of geometry could be adopted according to the goal of the experiments, like say shear flow experiment, stable gradient for soluble and surface bound factors for chemotaxis experiments, multi-layer fluidics for cell polarization and co-culture experiments. So, PDMS is the main line of thought or main material practiced in microfluidics labs, because it is easy to mold. Now, we are coming how to make this microfluidic channel using photolithography techniques, using a video basically. So, main principle is that replica molding. Replica molding is the basic principle is that you have a raised structure. Say here we are showing that structure, length is 2 millimeter, width 100 micrometer, height 50 micrometer on a silicon wafer. So, this should be means properly transfer that structure to elastomer. So, that is why, so this raised structure is prepared or done by photolithography techniques. After getting the raised structure, you can use that elastomer like say PDMS, then you polymerize it, make that solid form, then you peel it down, then you get that your channel structures. So, these whole the steps means up to this you are using that photolithography techniques, then the remaining steps are done, and all the techniques should be done in a very clean room, because it is the micron level, all the setups in micro level. So, that is chip design. So, what are the structure you need, that you have to design in AutoCAD or Clivins using software. Then mask preparation means your, that is your chrome mask generally you are using. Then spin coating of photoresist using SU8, exposure the development to make final raised feature, what we have discussed here, the mold on a silicon substrate, then master mold, pattern transfer to PDMS, say the pattern is transferred in PDMS, then you can means bonded by oxygen plasma method using your glass surface, and you punch the holes to make that ports out in and outside ports, then you can use for your cell culture in microfluidic system. So, we shall go through the video that your photolithography system. So, here that what pattern we have generated in that we are giving a laser writer to mask preparation, then we are coming to that spin coating with using that silicon wafer. So, this is properly placed here. Then it is properly balanced to make it is a proper alignment by which when it will be spinning there will be no unevenness, and we put SU8, then we spin it for around say 3000 rpm for 30 seconds and according to the need. So, after the spinning down we are pre-baking 65 degree centigrade for 2 to 3 minutes, then again it is baked 95 degree centigrade for around 2 to 3 minutes. Then we are loading the mask. So, first is your chrome, then in the top of the chrome you are using that silicon wafer with SU8 what we have done earlier. Then you insert it in the UV chamber, then exposing it for 2 to 3 seconds, by which that transparent part on that your silicon wafer it will be polymerized, it will be hardened, that unexposed part will be loosened, basically it is not polymerized. So, again you baked at 65 degree centigrade for 2 to 3 minutes and 95 degree centigrade again 2 to 3 minutes. Now, development of the your photoresist. So, this is SU8 developer. Here we can rinse it 2 or 3 times or several times, and then again you transfer it in isopropanol, again you rinse it 2 to 3 times, you will be seeing that all the white things are going out, then microstructure develops. So, to check that how the microstructure develops, you are seeing under the microscope to see that microstructure, say in our case it is a microfluidic channel, and if it is good it is fine. Next step is that on that what is the mask develops, we are using that PDMS as I mentioned earlier, 10:1 ratio, that base is to curing agent and is a very viscous like things, and you put it. Then some air bubbles will be entrapped. So, you have to remove the air bubble by suction, and that if air bubbles are there they can hamper the structure. So, air bubble are removed. Now, heating at 65 degree centigrade for 30 minutes cured the PDMS, now it is solidified. Now, you peeling down that your solidified PDMS with that your desired structures by using scalpel with some little bit precautions by which that structure should not be disturbed. Though it is solidified right now, not much disturbance will be there. So, this is your structures. Then we can say in our structure around say 8 channels are there. So, you can take you cut the each and every channel with the your dice, and you get that diced channels basically. So, this is the chip cutting step. So, you can see that this is that channel with that PDMS. Now, next step will be your plasma treatment by which it will be bonded to your glass surface. So, that is plasma bonding, we are entering that glass slides and that PDMS where that your channel side will be top, then exposed for desired time. Then after that you will be taking that plasma coated means plasma treated surface, then you properly aligned that surface which are exposed surface in such a way that that exposed surface will be coming together. Then you again you put some your PDMS in the sides to seal down, again you baked it, then you can punch it and get the structures. So, all these things are done by our research scholar students using our institute facility. So, next part of the microfluidic based cell culture system I shall be discussing in that next lecture, I think. Thank you.